My assigned protein was the Kras protein and had the mutation of glycine AA number 12, which is being replaced by aspartic acid, aka G12D. The KRAS protein is formed by the KRAS gene. The normal function of the KRAS protein is relaying signals from the outside of the cell to the cell's nucleus. The signals instruct the cells to grow and divide or to mature and take on specialized functions. This protein is in GTP phase and converts GTP into GDP. KRAS acts as a switch which is turned on and off by GTP and GDP molecules. It binds with GTP molecules to transmit signals, which converts to GDP molecules when deactivating transmit signals in the nucleus. The KRAS protein is produced by the KRAS gene, which is found in the 12th chromosome in the human nucleus. I believe the protein becomes more active because the mutation amplifies the GTP signaling pathways that leads to the development of tumors in the human body and mutes the GDP molecule signaling pathways. This leads to prolonged activation of its downstream pathways associated with cellular growth. The main functional group present in the original AA side chain is glycine. Glycine is the simplest amino acid with a single hydrogen bond. It can fit into a hydrophilic or hydrophobic environments due to its minimal side chain only of one hydrogen atom. Glycine has hydrophobic side chains that will exclude water associated with other hydrophobic side chains, while its hydrophilic side chains will bond with other hydrophilic side chains. The mutation of glycine at position 12 to G12D leads to the projection of a bulkier and negatively charged side group in its active site. This causes a hindrance in the GTP hydrolysis and impairs the GTP phase function. The mutation of glycine at the position 12 to aspartate G12D leads to the projection of a bulkier and negatively charged side group at the active site. This causes a steric hindrance in the GTP hydrolysis, impairs the GTP phase function, and locks the KRAS in its active GTP-bound state. Aspartic acid is a dicarbolic amino acid and has a polar amino acid with two carboxylic groups, and one of those groups is attached with an additional menthyl group. This is different from the glycine, which is hydrophilic and hydrophobic. The G12C mutation is a single point mutation with the glycine to cytosine substitution at codon 12, which favors the activated state of Kras. Because of this mutation, it amplifies the signaling pathways that lead to the development of tumors. The limitations of this model is the model cannot show the true function of the mutated Kras protein. The model cannot show how many bonds are actually in the protein, and the model cannot show the shape change of the mutated protein in real time. So this is my protein. This is KRAS, and it is the normal and mutated version. The normal version is with glycine, and the mutated version is with aspartic acid, or also known as G12D. So on this side, the little purple dots represent GTP, which helps the protein transmit signals to the cell's nucleus, which helps it multiply multiple times and produces GDP when it needs to stop. Now with the mutated version, the GTP doesn't stop and it continues to have the cell regenerate multiple times, which then mutes the GTP process. This is SW1, and then we have SW2 over here. Again, this is the GTP, and then right here is the P loop. And then on this side, as you can see, the protein is completely different now. because the mutation actually altered the protein. And so with this, it actually enlarged everything else. And so it enlarged the SW1 as well as the SW2. And then here is the P loop, and right here as well is with the G12D for the aspartic acid. And yes, thank you for looking at my presentation.